So for the female reproductive system, the purpose of the female reproductive system is to create eggs, provide the location where sperm arrives, and for the egg to get fertilized. And then once the egg is fertilized, provide a location for that fertilized egg to implant, and then subsequently develop and then clearly carry it to term. So the female reproductive system, as we see here, obviously we've seen a cross section from the male reproductive system that we did previously. So this is the female cross section. You should be comfortable naming the anatomy on either one of these male or female cross sections on your exam. Interesting thing that popped up on my Instagram a while back. So nice dress to wear out once in a while, you know, just showing your body parts off. Thought it was an interesting outfit. So the reproductive anatomy of the females, we're going to talk about the ovaries, and our progression today for our lecture is we're going to start from the ovaries, then we're going to go through the fallopian tubes, the uterus, and so that's kind of the plan that we're going to go through here. So as far as the female reproductive system, we can see in this image, again, the ureters, they're a little on the high side here. You can see also where the bladder is located here. Then obviously we have the urethra. So females, we just have one urethral segment, which is the membranous urethra. So you don't even call it the membranous urethra, just call it the urethra. Then behind it, this would be the vagina. Obviously, the uterus is here. You can see how the uterus arches over and sits on top of the bladder. The ovaries are here. So the ovaries are located in the abdominal cavity, just like we see the uterus also located within the abdominal cavity. But to hold them in place, we have this broad ligament. And the broad ligament is literally like a long, wide, thin membrane that is, it's like all these features that we're going to go through are laminated within that. This is the broad ligament. It moves across. It actually creates a membrane that separates the abdominal from the pelvic cavity. Within the broad ligament, we'll find others. We have the suspensory ligament that's coming here from the sides. Within the suspensory ligament that's coming on over, that's going to attach onto the uterus, that's holding it within the abdominal cavity, it also includes the arteries and veins. We also have a smaller ligament right within here that I'm circling. Those are known as the ovarian ligaments. It's connecting the ovary to the uterus itself. Otherwise, the ovary has nothing else to tether to. So the ovary lies within the broad ligament, but it's also anchored to the uterus through the ovarian ligament. Then we have this additional ligament coming out, and that is the round ligament. So we'll go through this here. This one is the round, suspensory ligament. We have the round ligament, and we have the ovarian ligament there. And then this one here is known as the broad ligament. Within the ovary itself, and this slide doesn't look good, meiosis, this is the one over here. So we'll put the Roman numeral here. Oogenesis means forming of the egg. The oocyte is the egg, ultimately. So for sperm, for males, it's going to be the sperm. For the females, it's going to be the egg, more professionally known as the oocyte. Oogenesis is the formation of this egg. Oogenesis begins when the female was an embryo in her mother. A female embryo has all of the eggs that she ever will have for her lifetime. The numbers drop some by the time that female is born. Then they drop a bit more by the time the female enters into puberty. The point here is that as opposed to males who then make sperm at the impulse from spermatogonia, you get follicle stimulating hormone, tell the spermatogonia, and you're making a brand new sperm at that time. Females have all the eggs that they're ever going to have from the time that she was a couple months old in utero. And then it hadn't quite completed all of meiosis. So meiosis ends with a cell that only has 23 chromosomes. Just like a sperm has 23 chromosomes, and when they get together, you'll have 23 pair of chromosomes. So in the females, because they are created so early on and they have to stay in this halted state of development for decades, it's a lot more stable for them to not have completed meiosis entirely. So they're really halted until about meiosis 1. And then from meiosis 1, instead of getting 4 cells, which really you get 4 cells, but you ultimately get one primary oocyte. That one's going to be potentially the egg that will become further developed. We're going to go through a series of developmental stages for these eggs and for these follicles is what we're going to be referring to them as. And even through the stages that we're going to be talking about in the ovary, they're not done with meiosis. By the time an egg ovulates and it escapes the ovary and is on its way down the fallopian tube, it is only at the stage of metaphase 2. So it's still not done with meiosis. Meiosis will actually be completed when the egg finally gets fertilized. The point that I want you to take from this slide is, first and foremost, the female's eggs that are in her ovaries were there since she was an embryo. The eggs have not yet completed meiosis. They are in a halted state of meiosis 1. And upon development that we're going to talk about today, meiosis continues until it stops at metaphase 2. So you should know that at the time that an egg is ovulated, it's still only in metaphase 2. And then it will be completed when it encounters a sperm. This picture we can see comparing sperm to an egg development through meiosis. They both start with one main cell. They both undergo meiosis. However, because the egg ultimately becomes a really large cell that has a lot of cytoplasm, in the meiosis process, one egg starts to, you can conceptually consider it robbing from its 3 sisters, if you will. So instead of getting 4 equal sweat cells like you would if it was in the male reproductive system, in the female, you end up with 4 cells, but really only one that's a viable egg, and it's going to have 23 chromosomes. This list are the follicular stages that we're going to go through as far as the development of one of these eggs up until the point it's going to be ovulated. This is a cross-section of a cat ovary, so there's going to be more follicles out there than there would be in a human. So if we have an ovary, we start off from your list with these primordial follicles, and they're way out in the periphery, and there's loads of them. These are the ones, these primordial follicles out here, that are in the female from the time that she is an embryo. The primordial follicles are going to be a cell that's ultimately the egg itself, or the oocyte, and it has these really thin cells, these little simple squamous cells around the outside. So it's real thin. We see, this is a close-up view of a single one, but lots and lots of them clustered throughout here. We have thousands of them. We go from the stage of a single primordial follicle. So I'm coloring the egg as red. We want to keep track of what the egg is versus what are the different supporting cells around it. On a given cycle, about 10 to 20 of these primordial follicles kind of emerge and start to develop. And they become primary follicles. These primary follicles are still, the same size as far as the egg goes. But these cells have the outer cells that start to grow, these little cuboidal cells that get a little bigger and more plump as they're developing. They go from a single, these are more primordial follicles right here, and then we're moving into a single primary follicle. That's the next stage. So the primary follicle is still just the egg. Now, instead of a thin, simple squamous layer of cells, these turn into little cuboidal layers around the outside. They go from a simple layer of one layer And then they can, move over and become stratified. And it becomes a thicker layer of cells on the outside. And in fact, some of these cells start to develop. So I'm going to add another color. So these are primary follicles. The primary follicles, again, they're 10 to 20 that emerged from the primordials. We still have thousands of primordials left, but we bring 10 to 20 forward. Some of them maybe don't come out of their meiosis, stasis so well, and so they don't really develop. So it's really just seeing which cells are going to develop, which ones are going to start to respond. And so the energy of the ovary starts to become directed to the more viable ones. So we can see that they're either going to be simple, they start off as simple simple cuboidals, and they get a little thicker, stratified cuboidals, but still we have the egg inside the oocyte. In this picture, we can see several primordials, again, out by the periphery, and then we see a few primaries here that are now having had emerged. Then we go into secondary. The secondary follicles are, again, with the egg. The egg is still in there in the middle, but what we start to do is we develop this fluid-filled antrum. Antrum is the fluid space. And then ultimately, it starts from the secondary where the fluid-filled antrum just begins to separate away from the egg to a mature follicle where you can see a significant antrum, but the egg is still right here. Draw this. We still have a few cells right around the mediate part of the egg. And then we have more cuboidal cells that are kind of coming around. So this here, and we've got some of these other green ones I'm drawing. So this would be a secondary. And then ultimately we come to a mature or Graafian follicle where the antrum just enlarges even more. Just drawing lots of cells. I'm just going to make them squiggles. And we have some of these other cells over here too. So we have primordial, then we have our secondary follicle, which is going to be there. And then we have a mature, or known as a Graafian follicle. And they really just look, the only difference is the mature one has a larger antrum. And so it is really the development of this antrum that's going to help it when it finally becomes ovulated. One thing I'm going to point out here. So I want you to know about what part is going to stay in the ovary, what part is going to be on its way. So ultimately, this is what's developing within the ovary. We start with the eggs out here in the primordial follicles. Then the blue cells, the tissues coming around, are going to be what's going to be the follicle itself. Think of it as the shell or this bubble that the egg inside is going to be developing within. So we have these cells that start to enlarge, but what ultimately leaves the ovary when it's time to ovulate is going to be this that I just circled in blue. Draw over here what the anatomy of that egg is. First we have, this is the egg, this would be the oocyte. And then we would have a layer just around it that's protecting it on the outside. And this here is called the zona pellucida. The zona pellucida is a sort of a shell of the egg, and then outside of that are just a whole bunch of cells that are protecting the egg, that will be protecting the egg as it travels down the fallopian tube, and those are known as corona radiata. So what ultimately leaves the ovary at the time of ovulation is the egg inside, its zona pellucida that's immediately around the egg. We can usually see it as a sort of a definitive line around the egg itself, and then a cluster of cells that just hang on around the outside of that. The two layers that a sperm has to penetrate to get to the egg, to fertilize it, is first it has to make it through these kind of loosely covering cells, the corona radiata, and then it has to penetrate the zona pellucida. Who remembers sperm anatomy and what part of a sperm is going to be utilized to penetrate that zona pellucida? Acrosome, excellent. Let's just do a little quick drawing here of a sperm. So if we have the head of the sperm here, where the nucleus and the DNA, remember we have the mid piece and the tail, and then sort of this helmet portion of the sperm is the acrosome. That's what's going to have to penetrate the zona pellucida so that the head of the sperm with the nucleus, with the chromosomes, can actually enter inside the egg itself. The corona radiata are protecting this egg as it's going down the fallopian tube, because one thing to remember or realize that the fallopian tube is not like an open tube like a straw. So it's not like a kid going down a slide, you know, one of those tube slides. It's really kind of more obstructed than that. And so the egg sits there and it's moved along by fimbriae throughout the fallopian tubes. It's got to really be maneuvered through. The egg's not going to move through on its own. You don't want that touching the egg directly. So the corona radiata really protects. In this picture, we can see really nice primordial follicles out near the periphery. We see a remnant primary follicle here. Don't see a secondary follicle, but we have now a mature follicle. We can see the egg with its little pronucleus inside. See this darker pink line, a very distinct line. That's the zona pellucida. And then we see some corona radiata cells as it's really docked over here. So this is migrating to the edge of the ovary, and ultimately pressure developed in here is going to propel and eject just the egg itself with the surrounding 2 layers of protective layers, and that's what's going to get launched. So then we're going to talk about what happens in the ovary of the leftover. The follicular cells way out here that's forming the follicles, those stay in the ovary after ovulation, and they then transform and play an important role. Okay, so far we've got ovary. We have primordial follicles that are in the ovary from the time the female was an embryo. After puberty, hormonal levels will instigate development. It'll be follicle stimulating hormone. We'll tell a few of these, approximately about 10, to move out and go from their primordial follicle stage into a primary follicle stage. Then only a few leave the primary follicle stage. We're down to about 3 or 4 at the most. They become secondary follicles, and antrum starts to develop. Not all of them will progress. And then one finally ends up becoming a mature or Graafian follicle that then migrates to the edge of the ovary. And then a couple days before, there's actually estrogen levels are rising this whole time. It starts at low levels. Estrogen levels begin to rise. The rising level of estrogen ultimately feed back to the pituitary gland to give that luteinizing hormone surge in mid-cycle that's actually going to cause the egg to be ejected. And the reason why I keep using the term ejected is that the fallopian tube, where the egg's going to end up going to, isn't really attached to the ovary. It's sort of like over here, and here's the ovary. And there is this literal leap of faith that this egg is like, once it gets launched out like a little catapult, that hopefully it's going to make it into the fallopian tube. And sometimes they don't. They wing off and will go into the abdominal cavity and then just be absorbed and go away. So at this point is where we have our Graafian follicle bringing it to the edge. We have the end of the fallopian tube, and we're going to see these are fimbriae. So that fimbriae to kind of wrap around to try to catch this ejecting egg. So we go from this point to the egg actually launching out, going in. This had moved over in order to catch it, but we have the leftover cells in. So during this time, we were developing, estrogen is rising, but we haven't mentioned progesterone yet because it's not there. So these cells here that are left over, once the egg has vacated this mature follicle, these cells become completely enormous. And this puffs up into a very, very large follicle. And the walls get really large, And what they do is this ends up, they secrete progesterone, but this is called the corpus luteum. And the main job of the corpus luteum is to secrete progesterone. So I want you to notice with this image, once the egg is on the way to the uterus, it's left the ovary, from the point of ovulation onward, progesterone is where it starts to secrete. So you're not going to see progesterone levels in a female in the first couple weeks of her cycle. Only after ovulation has taken place will progesterone be present. It's good to know clinically, if you're going to have hormones evaluated, it's more important to have your hormones evaluated in the last 2 weeks of your cycle, because it's not so important as what your levels are. It is more about balance, not about absolute levels. So we could measure all the women's hormones in this class. We might have some women with hormone levels about this high, and we have some women with estrogen levels about this high, and we might have the estrogen levels that are really, really high. That by itself doesn't make much of a difference unless it matches their progesterone. When progesterone finally gets kicked out, if you have this much estrogen, you're going to want this much progesterone. If you have this much estrogen, you're going to want this much progesterone. So it's the ratio of the two that they balance more than the absolute value, which is why the range of what's normal for someone's hormone levels is pretty wide. And someone could be smack in the middle and have symptoms of too much or too little, because that would be maybe too high or too low for them. But it's still in the normal of the range. When you measure 1,000 women, this is what the range could be, but it doesn't necessarily mean. So evaluating hormones can often be a tricky thing. This is a picture from my old textbook. This is where you can see the corona radiata quite nicely emerging from the ovary itself, but you can see the gap. There is a gap, and it's this little leap of faith that this egg is making to make its way into the fallopian tube itself. Here we can see the corpus luteum. So this is an actual ovary. An ovary is probably, as big as the end of my thumb. So if you were to take your thumb and nip it off, that's about as big as someone's ovary is. They're a lot smaller than people realize that they are. And so you can see it with the naked eye. This one's you're looking at a little close, but this here is a corpus luteum. So you can see how huge this is. It becomes literally a progesterone gland. The trick here is it secretes progesterone. That's important. It secretes progesterone only after ovulation. So that's your second most important point. And then the third important point that you need to know is that it only lasts for 14 days. This is one of the finite hard and fast rules for the female reproductive system. So female, normally we talk about a 28-day cycle. So 14-day progesterone will actually just happens to correspond with day 14 ovulation. You add 14, you're 28, then you're at the end of the cycle. So if a female has a longer cycle, say 54 days, it doesn't have to be a bad thing. It just is a different cycle length. But because the corpus luteum only lasts for 14 days, a female that is a 54 cycle day length would have ovulated on day 40. You just end up subtracting 14 days, and that's where the person ovulated. So the most variable time in a female's cycle is really the early stages. Is it just not, taking a while to get the development started? Once the follicles start to develop, ovulate, everything runs just like clockwork. It's really just getting it up and going. So that's where the variation can be. Unless someone's at the tail end of their reproductive life and they're starting to enter perimenopause and menopause, they might start having sort of shorter cycle lengths, but often it's because they're not ovulating and they're actually not creating a corpus luteum, so they can have a short cycle for that. But that's for here for another time. So generally, the most variable time of a female cycle is going to be the early follicular phase. So developing all these follicles is known as the follicular phase. Once the egg is left the ovary, it will now, and the corpus luteum is present, it is now in the luteal phase. So there's only two phases that you need to be aware of for the female reproductive system's ovary, is the follicular phase and the luteal phase. We're going to learn some other phase names with regard to the uterus coming up. This is a histological slide showing the corpus luteum itself. At this point, you can see how big it gets. Now, the corpus luteum, as I mentioned, lasts only 14 days, so it's 2 weeks long. When at the end of 2 weeks, it stops churning out progesterone. So it's been sending out progesterone, doing its thing. Your progesterone levels will rise. You can do a blood test or a saliva test to evaluate it. And then after 14 days, your levels suddenly drop. It is that rapid drop in hormone levels that will then initiate the release of the endometrium and initiate menstrual period. So we'll talk about the endometrial side with this, but it corresponds and is initiated by the presence or absence of the corpus luteum. The point that we're at here is we start from primordial, we develop, we create an egg, launch the egg, it's on its way, which we're going to talk about the fallopian tube in a moment. So the whole thing that we're going to talk about now with the fallopian tube and if the egg's on its way, remember, back up at the ovary, we have a corpus luteum just churning out progesterone. The progesterone gland, that's what it's doing. So the fallopian tube, also known as the uterine tube, is where the egg has been launched into and is going to travel on its way to the uterus. We have you can see the ovaries. And so we start off with fimbriae. Those are the finger-like projections that are kind of wrapping around or near the ovary. But they don't touch it, they don't hang on to it. There's a gap, and so there is this space and it's sort of hovering around. But the fimbriae have this job, because they're finger-like projections, they kind of hang out over here, and the closer to the time of ovulation, there's a signal mechanism going to the fimbriae that actually brings the fimbriae around closer to the ovary, and they start to undulate in anticipation of an egg getting popped out. It could get popped, ejected out through a side. And so these finger-like projections are undulating to try to capture this egg and bring it into the fallopian tube so it doesn't, wing out into the abdominal cavity. So we've got fimbriae, I mentioned here. Those are the finger-like projections. The infundibulum is just the portion that the fimbriae project from. The other important part to notice is the ampulla. The ampulla is really in this arched area. It's in this arched area. That is the most common site for fertilization. Now, it's important that fertilization take place way out there because a fertilized egg has to be developed about 5 to 7 days before it can implant into the uterus, the endometrium. So first of all, an egg is only viable for about 24 hours at best. So it's really not going to last much farther than the ampulla. In the freakish scenario that an egg makes it all the way into the uterus here and gets fertilized, even if it gets fertilized there, it wouldn't reach a sufficient level of development to even stick to the wall and stay. So it's important that we get fertilized way out in the ampulla. That allows not only time for development to take place before it arrives into the uterus, but it also allows the uterus time to prepare for the fertilized egg to arrive. So this is what a cross-section of a fallopian tube looks like. And I point this out, we're not going to see this on an exam. I don't have the histology of it. I just want you to see it to realize it's not an open tube. It's not like a straw where just an egg's going to roll right through like a tunnel. It's going to be about, this big. So it's not really going to be rolling through on its own. It really needs to be helped moving along. So this is important, and it's also why the egg is going to need its protective layer, that corona radiata on the outside.
The reproductive cycle begins with ovulation, the release of a ripe egg or ovum from the ovary. Hundreds of thousands of follicles sit nestled inside the ovary. Each follicle is a sphere of cells with an immature egg at its center. The typical 28-day menstrual cycle begins on the first day of menstrual bleeding. During the first 7 days of the cycle, a few follicles start to grow. These maturing follicles secrete estrogen hormone into the bloodstream to prepare the lining of the uterus for pregnancy. Around day 7, all of the follicles stop growing and begin to degenerate. except for one. This dominant follicle continues to grow, nourishing the developing egg inside it. Around day 12, the follicle secretes a surge of estrogen into the bloodstream. When the estrogen reaches the hypothalamus and the pituitary gland in the brain, the anterior or front part of the pituitary gland floods the bloodstream with luteinizing hormone. Around day 14, luteinizing hormone causes the follicle to undergo a sudden growth spurt. Right before ovulation, the egg detaches from the inside of the follicle. The bulging follicle releases chemicals, signaling one of the two fallopian tubes to move in closer and envelop the follicle. The follicle swells until it bursts open, ejecting the ovum and fluid from the follicle into the abdominal cavity. In response, the fimbriae, tiny finger-like projections at the end of the fallopian tube, sweep across the ovulation site and pick up the ovum. Microscopic cilia on the fimbrial surface guide the ovum to the entrance of the fallopian tube. Muscular contractions along the wall of the fallopian tube gently propel the ovum toward the uterus. The ovum lives for 12 to 24 hours, the interval where fertilization can occur. If not fertilized, the ovum will disintegrate and be shed along with the uterine lining during menstruation, marking the end of one reproductive cycle and the beginning of another.
So this image here, you can see actually the fimbriae, the fallopian tube. Here's a 3-year-old, 14-year-old, 27-year-old. So there's a couple things I want you to notice from these slides. First of all, the fimbriae, you can see the arched fallopian tube, but also the ovaries. So you can see the ovaries here, but here's a 27-year-old, so it's not an older ovary at all, but you can actually start to see that there are some pock marks on the surface of the ovary, and that anytime a follicle is ruptured and it's ovulated, it actually creates a little scar on there of where it had ruptured from. So you can see from not having any ovulation to already having some, although it's not a really clear picture. So then from the fallopian tubes or uterine tubes are attached obviously to the uterus. So we'll get now to the anatomy of the uterus. Here's the fundus of the uterus. Obviously the body is going to be the main part, and the cervix is going to be the part of the uterus that's projected down into the vagina itself. So the cervix is really the gateway or opening into the uterus, or the blockage to prevent anything from coming in. So normally the cervix is closed it's like round, like a donut, but closed tightly until it's near the point of ovulation, where it becomes a little less inhospitable. It will actually soften and open slightly, and the prostaglandins in the seminal fluid will also help to contribute to that as well, to allow some sperm in. But the cervix that's down here, it's within the vaginal canal, but it's the cervix that's getting sampled, the cells of the surface, when someone's getting their pap smear. So who had done their prerequisite class, and you've done like a cheek swab, and then you looked at your cells in lab doing something like that? That's pretty much what a pap smear is. You just have a really long Q-tip going in there, and you just swipe it just like you would your cheek if you're doing that lab activity, but you're swiping a cervix. And the reason why for females the cervix needs to be evaluated is that on a cyclic basis, but it's changing on a monthly cyclic basis, and that it's either going to be really close or it's going to be, so it's responding to hormones. And because it can respond, it could be a location where mutations could occur, and that those mutations could be continued unchecked. So you get a pap smear, make sure that the cells in there are normal and not susceptible to mutations or having already started mutations. So that's why it's important that females get a pap smear. It's a super simple process. You just get a swipe of it, and then they just look at the cells. Is the cell normal or not? On a little health note here, if a female, so there was a study done, and that had a group of women that went to their OB, got their pap smear, and it's the females that had not a normal pap smear, but it really wasn't abnormal yet. So you didn't really see any pathologies. It wasn't a cancerous or anything, but they definitely weren't normal. So you have this population of females. The study was, they divided them in two groups. One just did their normal everyday thing, waited their 6 weeks and went back to the doctor and had another pap smear. The other one, they started to take in high levels or higher levels of folic acid. The group of women that had higher levels of folic acid just for the 6 weeks until they had their pap smear had a significantly higher number of pap smears that were like, oh, sorry, they're normal, don't worry about anything, and that had regressed back to normal. So the cells that were starting to change but had yet to become pathological came back into normal territory, where the other ones were more of, let's just keep watching you, make sure, you know, might have to have more frequent pap smears just to make sure nothing's developing. So, but that's at this level of the cervix. So the cervix is a little more dynamic and has changes, and that's why it could potentially be susceptible to cancer. You should know, as far as the ligaments go, we have the uterosacral, or we know the sacrum's in the back. So if you have your uterus, uterosacral, like tethering it to the back. None of the images I have show a uterosacral image, so that's why I sometimes skip it on the list. The round ligament holding it to the front. You have a basic round ligament that's going from the uterus more towards the front of the pelvis. Then we have the suspensory ligaments that are coming in from the side. The suspensory ligaments are the ones that also have an artery and a vein, the uterine artery and the uterine vein. And then we have the broad ligament, which is that wide, flat one that's going to laminate everything in together and kind of hold it in place. The layers, or the wall of the uterus, we have the outermost layer. The serous coat is really just the outside that you see, it's just connective tissue. Then the thickness, the wall, the vast majority of the wall of the uterus is going to be the myometrium. Myo means muscle. So the myometrium here is going to be the thick portion of the uterus. And then the endometrium is what we're going to be focused on from here on out as far as the uterus goes. The endometrium is what's going to be changing throughout every monthly cycle. The myometrium just stays the same, so does the serous coat. So the endometrium is going to be our variable stage. Estrogen is going to promote growth. Progesterone is going to promote secretion. So they both are involved in the development of the endometrium, but they do different. And we'll go into the details of the estrogen and the progesterone as we go on. So this shows more of a time lapse, the cartoon portion. It's in, well, what they're saying is this is the menstrual stage, and then the following week it looks like this, and then the following week it looks like this, and then it goes on. So it's a continuum across a month. That's what the cartoon depiction is. What the histology slides are is during the menstrual phase, this is what it looks like. So there's really no endometrium. This is the end of the menstrual phase. This is actually the myometrium, the muscle that's going to start below. Then during the rebuild phase, which is the proliferative phase, you can see the endometrium starting to grow, and we see these glands starting to develop. During the proliferative phase, the hallmark view that you're going to get is really the glands are really geometric in shape. The glands are going to be like round, and then as it grows thicker, they elongate and they become oval, but they really have really nicely defined borders. So that's really what you're looking for when you see a slide. This one, not as much, but I was looking for some that were more size matched as far as the zoom in level. This last one is the secretory phase, which means it's secreting. So if you're building glands here, but it's here that the secretions are actually taking place. It's now functional. So just to get ahead, proliferative is the growth phase. This is going to be dominated by estrogen. The secretions, this is dominated by progesterone. And the menstrual phase, how we get to the menstrual phase is really after the level of secretion. Remember, if it's about progesterone, remember how long does progesterone last from the corpus luteum? 14 days. So on day 14, all of a sudden, this was getting progesterone. Day 14, it's not. So it has no progesterone. That's a signal for the cells to start to disintegrate and start to detach. And it literally starts to crumble and fall apart. And then pieces of it are falling out of the uterus, sort of like a slow time release avalanche. And so what you have is what was a thick secretory layer that crumbles down, and now this is the end of the menstrual phase. So sometimes if people were looking in class here and we're looking at menstrual phase histology slides, students often say, but it looks just like the secretory phase. And it really is. It's at the secretory phase development, but then it's just falling apart, really is the only hallmark there. Why would week 1 be the menstrual phase when you're just getting rid of the old endometrium? And so it always seems backward. But the reason why week 1 is considered week 1 is because that's when, although I erased it, the new primordial follicles are becoming primary follicles. Week 1, it could be 1 day, it could be 2 days, it could be all 7 days. So the menstrual period varies considerably from female to female. So it's sort of considered week 1. That's why there's a bit of a crossover. So when I talked about 1.5, it's just whenever the period is done, that's it, is going to be the proliferative phase. And then you're going to rebuild. So this is the muscle part. We can see we're rebuilding this endometrium. We can see the glands are really just circular, more geometric. You can see here, they're starting to stretch out, starting to get thicker. These are the new glands here, and that's pushing upwards. So these are very proliferative looking endometrium. And then after week 2, remember that's when then on day 14, ovulation takes place. And then we have the corpus luteum, which sends out progesterone. So when progesterone hits this endometrium, that's where the glands start to get really squiggly and they start to actually become active. So the glands in the endometrium start to churn out its product, and its product is a glycogen-rich substance that if a fertilized egg were to make it into the endometrium and hit the wall, it burrows under into the wall, so it sort of migrates under, and it taps into these glandular pockets. And these glandular pockets will have to continue secreting their glandular substances until a placenta can get made and they get the nutrients through the blood. So that's going to be months. So the secretory phase is really important in that you have a viable endometrium for fertilized egg to implant. So not only, so the proliferative phase is the making of a new endometrium, and it's more like scaffolding, kind of the framework of a house or framework of the endometrium. Progesterone comes in after ovulation, so it's these last 2 weeks that take place that make this now the secretory phase, where it's secreting all these nourishing substances. Now we know it lasts only 2 weeks because if no implanted embryo, so no implanted fertilized egg, so early embryo by the time it gets in there, gets implanted, then that's when the 14 days it'll be released and then you start all over. Now if there's an implant, something implants in there, it changes it. And that will be what we'll talk about when we come back. So the vagina is just really a tube, a muscle-lined tube, semen's deposited, menstrual flow exits that way. It's a channel. It tends to be really, really acidic, which damages sperm. Closer to ovulation, you become a little less acidic and a little more hospitable or a little less offensive to sperm. There are mucous membranes all along the channel. Hymen is periphery of sex. There's a barrier, but it doesn't always there. It's not always intact in people. The more active a girl is when she's young, hymen can just be no longer there or no longer intact. It doesn't always indicate sexual activity. The external genital organs. So the vulva is really sort of the outer genital organs as a package. The vestibule means it's just the opening space. So you have sort of the vestibule is the space within here. We have this portion here we always ask. It's just the symphysis pubis. So it's the bony portion of the pelvis, just so you have a reference point. So then just below it, this here is the clitoris. The clitoris is actually made of corpora cavernosa. It's made of erectile tissue, the same stuff that a penis is made out of. So we have very analogous tissues that are just sort of arranged in different ways, males versus females, which is why in some problems with development, you could have ambiguous genitalia, you can have hermaphrodites, there can be a lot of variations and changes in there because we are really starting with the same basic tissue types. We just assemble them differently, whether it's going to be male or female. So here's the clitoris. We have the urethra going to the bladder. There's the vagina going to the cervix, which is the bottom of the uterus. The cervix is a part of the uterus. It's just projected into the vagina. Fallopian tube going out, ovary, the ovarian ligament there. The rectum is right here, which is a place where women can get a digital exam to have their cervix felt. So if they have a pap smear that would suggest perhaps they have abnormal cells, then the next logical progression would be the doctor would do a rectal exam on the female, just like they would do on a male. If you're checking the prostate, you want to see how big is it? Is it bumpy? Does it have abnormal growth? Same evaluation is really going to be done on the cervix for the female. But it's thin-walled, so it is very clinically convenient to access the cervix and determine if something's wrong with it. And it's important to have that evaluated, as well as we have glands within, obviously, mammary glands, breast. So the mammary glands that women have, size is fat dependent. So fat content of the breast determines size. So whether a woman has like size D cups or size A cup, their lactating potential is the same. So just because someone has larger breasts doesn't necessarily mean they're going to produce more milk than the next woman. It's going to be superficial to the musculature. It's going to be on top of the pectoralis major. Within breast tissue, you're going to have these glandular lobes with ducts in them. And these ducts terminate in these enlarged areas known as lactiferous sinuses, sort of the spaces where milk is collecting. So a female that's lactating, has a baby that's been nursing, will have, obviously, will be on a cycle that usually matches the hunger cycle of their infant. And so their production, whether it's going to be high production or low production, obviously, the milk will then collect here in the lactiferous sinus. And then if all of a sudden maybe the baby's taking an extra long nap, but it's normally their feeding time, the female could have enlarged breast. It could start leaking even because it's on that cycle. But the milk hangs out in the lactiferous sinus. It's prolactin that stimulates the breast tissue to generate and create milk. It's oxytocin that's going to actually contract on the smooth muscle along the ducts that's going to then deliver the milk downward into the lactiferous sinus. And it's also then oxytocin that responds, like when the baby's crying with regard to hunger, that the mom will actually have what's known as milk ejection, and milk will actually come out of the breast and facilitate the baby drinking the milk. And so that's the difference between oxytocin, which is more of a delivery mechanism, versus prolactin, which is a glandular stimulant. I put these colors in, notice the green and the blue. The green are the stages that's termed from the perspective of the uterus or the endometrium. And the blue are the ovarian stages. So we can see that we start off with the menstrual phase, but in the pre-ovulatory time, in the ovary, it's the follicular phase. The follicles are developing at the same time, really, the menstrual phase happens, and then also while the endometrium is rebuilding. But once the egg has launched, we know the ovary is in the luteal phase, progesterone's present, which makes the endometrium secrete. So that's where it's sort of put here. I don't necessarily think this is the best way. I'll do it on the board again. Okay, so let's start here. If we do week 1, in week 1, we'll talk about the ovary first. In the ovary, we have, what's going on? We have our little primordial follicles. Week 1, we have our primordial. They are going to become primary follicles. So we have our little primordials. I should draw them in red. Then they become the primaries. It's a little bit bigger. But then those primaries have the cuboidal cells around the outside. And then in week 2, the primary follicles become secondary. And those secondary turn into mature follicles. So that's where we have our little eggs here. We start off with the cells around. We get a little, start to get an antrum, then we get an even bigger one. Push these out of the side. so that we have a mature follicle that's coming here towards the edge. So it's gonna be week 2. So at the end of week 2, the mature follicle, so this is, we're gonna say day 14. At day 14, that egg is going to launch out. So we're gonna say day 14, ovulation, and the egg has been ejected from the ovary, and now we don't longer have it inside. And it goes out with its outer barrier, the zona pellucida, as well as its other cells, the corona radiata. And so we'll do a little Fimbriae here into the fallopian tube or uterine tube, and it heads on its way to the uterus. Now, meanwhile, after day 14, so that's day 14, so then day 15 is the start of week 3. Now we went from a mature follicle to now egg is out to an empty follicle. That empty follicle puffs up, and what it does, it becomes the corpus luteum. So that corpus luteum is here, and it enlarges and gets really huge. And this corpus luteum is going to do what? Yes, secrete progesterone. So it's going to make, and the important thing about its progesterone secretion is that over here, we're going to do a stumpy fallopian tube. And then all of a sudden we have, here's a uterus with a really short fallopian tube. And so what happens is, and I didn't talk about the development yet, but that progesterone is going to then target this endometrial lining and to make it actually secrete and activate the glands. So that's really from the perspective of the ovary, what's going on. And so in the corpus luteum will continue for 2 weeks. So we have 14 days, which is unfortunate that it's day 14 is ovulation, you have 14 days. So just know it's 2 weeks long, the progesterone goes, and then after day 14, it's cut off, progesterone doesn't secrete, and this then goes down and atrophies and goes away. And you have a sudden drop of hormones, and then the endometrium is going to break apart and go out. So that's the big picture from the ovary side. So when we talk about the phases, this first 2 weeks is known as the follicular phase. And the last 2 weeks is known as the luteal phase. Week 1 and 2, follicular, and then 3 and 4, luteal. Done those in different colors. So then we'll come back. I'm going to erase this uterus and then do the uterus side because I skipped some steps, right? Okay. So we're going to start over, but then we're going to talk about the uterus while we're doing this. But we'll still use the ovary as our time because that's really what's dictating week 1 and 2. So again, with the week 1, we have our primordials becoming our primaries. During week 1, the uterus over here, so internal os, used to have a whole endometrium, but week 1 for the uterus, here you go. Week 1, it's going to be the menstrual phase. And so what's happening is this endometrial lining is breaking down and going out. So that's our period. Then week 2, we'll just to round it off as week 2. It doesn't have to be all 7 days. It could be 1 day. It could be 2 days. And then now you're looking at, day 4 through the next week would be proliferative phase. So during the proliferative phase, where this endometrium now has lost its lining, there's just a couple cell layers thick, the lining is going to rebuild, and these little glands are going to be more circular. You know, these glands over here, real geometric, just real angular looking. It's during this week 2 that the secondary follicles are becoming mature follicles. So again, we get the mature follicle, the cell, we have a few cells around it. We've got corona radiata, a couple cells around it, and then you have this huge follicle, these cells here. So week 2, we have the secondary becoming a mature follicle. We have the endometrium rebuilding, glands are being created. They're not functional, they're just being made and formed. And then we hit the day 14 ovulation, where the egg is out. There's the egg, has its zona pellucida layer, has its corona radiata around it, and now it's on the way. On the way to the uterus. And what's going on here is now our leftover follicle component inside is now, we have the corpus luteum. Here we go. Corpus luteum. It's making progesterone. The progesterone is now feeding over through the entire systemic bloodstream, not like a direct current, but it's throughout. This is getting secreted out through the mom's ovarian vein, coming back, and is now circulating. So now progesterone's going. And so what's happening is inside these glands, not only did this endometrium get even thicker and there's more glands, but now these glands are becoming active, and these glands are creating pools of a glycogen-rich substance, so that if this little egg that's on its way gets fertilized, it now has some nutrients to tap into. And so this is lasting for 14 days, giving a chance for the egg to get fertilized, get adequately developed, get stuck into the endometrium, and give a signal to not have it be released. And that will be the topic so when we come back. So what we have going here is, once we have progesterone, we have week 3, and it's known as the secretory phase. And it continues into week 4. So we go from these weeks 1, 2, 3, 4 with those names: follicular phase and luteal phase. The ovary is pretty easy. You're either making the egg or you're turning out progesterone, follicular or luteal. We add a little layer of complexity because now we have the same weeks with different names, but it has to do with what's going on in the uterus. But then instead of having 2 just across, halfway across, we've got the menstrual phase, which gets rid of the old endometrium, rebuilds structurally a new endometrium. But when the corpus luteum is present with progesterone, now we have a secretin where we have a functioning active endometrium. This lasts for 2 weeks, waits for the egg to get fertilized. No fertilized egg shows up. That's when the 14 days, this goes away, drop in hormone levels, and the endometrium will crumble. This is what they always show you in the textbooks. In here we can see starting, so this is how they're showing it. Starting from a primordial, which is out here. You go to the primary, secondary, Graphian, or mature to where you ovulate. So that's what we were drawing here to where we ovulate. Now the egg is on the way, and the rest is this corpus luteum secreting progesterone. So you can see in the black line, you have no progesterone until ovulation hits. And now that the corpus luteum is active, progesterone levels rise significantly. Now they are not to scale of the estrogens. Again, they're about 100 to 1 as far as progesterone to estrogen. And you have a huge amount of progesterone compared to estrogen. So you're going to need to know the weeks. What are the phases from the ovary perspective? What are the phases from the uterus perspective? And then what are the dominant hormones? So the early weeks, when we have those follicular cells here, the cells around in this mature Graafian follicle, I'll put the egg back in here, the cells here. These cells out here are known as thecal cells. And there's other granulosa cells too. They make estrogen. Estrogen is the growth. So estrogen promotes growth. That's why you have the proliferative phase, because it's growing and developing and making a new endometrial lining because of estrogen. Estrogen and progesterone have a relationship where it's like a yin and yang. One's going to be the growth promoter, and then the progesterone is going to be more of the mellow out, not so aggressively growth. That's why estrogen is often promoted or associated with cancers. Progesterone in balance will then have the growth, temper the growth, and turn it into secretions and function, and that's more of a progesterone issue. So when we talk about dominant hormones, before ovulation takes place, estrogen is going to be the dominant hormone in terms of growth and development. After ovulation takes place, it's all about the progesterone doing secretions and for getting this uterine lining ready for a fertilized egg. Women that are on birth control for many years will suppress ovulation. Obviously, they're trying to prevent ovulation and prevent growth. So if you suppress ovulation, you don't ovulate, you don't have an egg leaving, what hormone will you become deficient in, or what will you be missing? Progesterone. So women that are on birth control pills will actually have, not as much progesterone. Okay, not a big deal, because you don't really need an endometrial lining. You have no implanted egg that's going to go there that you want to go there. That's why you're on birth control in the first place. So that's not a big deal.